I am inviting Dr. Archana Mishra, a senior resident in Department of Pharmacology, AIIMS Bhubaneshwar. Ma'am has pursued her MD Pharmacology from AIIMS Bhubaneshwar and DM Clinical Pharmacology from AIIMS New Delhi. Her areas of interest have been psychopharmacology, neuropharmacology, clinical trials, and pharmacovigilance. Ma'am has received Young Investigators Award on 14th World Congress of Biological Psychiatry, Vancouver. Ma'am has published over 26 research papers in various highly reputed journals. Ma'am will now enrich us on the topic sample size collection. Over to you, ma'am.
Thank you so much. Hello, everyone. So over the last 3 days, all of us have been listening to many things, many aspects of sample size aspects of clinical trial, the management, data management, analysis, and all overview, everything. So now to start with, in a clinical trial, we should know how to calculate and basically what is a sample, what is the requirement of a sample, and why do we calculate it and how do we calculate it. So basically, we will interact over the next few minutes over this thing of sample size calculation. So when we have done our clinical trial, basically we start our trial with an objective of getting some results, right? So when we have started our clinical trial, we go through the entire process, which we have been listening in last 3 days, and then finally we get some results. So while we are interpreting those study results, those numbers, they have to be valid, they have to be reliable. That is the whole exercise is based on. So validity is basically having measured what we wanted to measure. So basically it would be very close to accuracy, as in what we wanted to measure and what we have measured are exactly the same. The second thing is reliability, that if someone repeats this experiment with our protocol, the person is ought to get the same results as we have got in our trial. So these two aspects are very important, two pillars of, clinical trial results. Now, how do we say that it is valid? So for validity, there are two aspects. There is an internal validity and there is an external validity. Internal validity means that whatever you have written in the protocol is correct and you have followed it step by step. Your methodology was correct. And that is why you can say, because we followed everything correctly, our results are valid. The second thing is external validity. That is how generalizable your result is. How can you generalize that to the entire population? You have taken a sample and now you have to tell that this results reflect whatever is happening in a population. So over the next few slides, basically, I'll try to tell about population versus sample, how much is enough, and significance of optimal sample size, and how do we calculate that sample size. So now we have various types of studies. You would have been listening to different classification of types of studies. Here, I'll go with a qualitative and a quantitative study. So we are mainly concerned with quantitative study here. And in that quantitative, we could have something exploratory, like we have some notion in our mind and we just want to have a proof of concept, and we are trying to explore a few things. In these studies, basically, we don't exactly calculate sample sizes. We try to talk about feasibility, and whatever is feasible, we take those number of patients or participants, and we continue with our study. The second thing is confirmatory studies, which are basically phase 3 studies, academic trials, and all those things, where we want to confirm a proof of concept, which we already have. And in those cases, we go for an optimal number, which we want to test that intervention on. So now these are the two, excerpts from two articles. The first one says efficacy and safety of oral isotretinoin monotherapy versus combined therapy with picosecond laser for the treatment of acne scars in Asian population. The second one says 12 months follow post-market study of a hydrophobic intraocular lens using a preloaded automatic injector in an Indian population. So do you think, is it possible, is it humanly possible to work with population? We can't go ahead and include all the population for whatever study or whatever trial we are doing. So now the question is, if we can't work with entire population, then how much is enough? Is it 5? Is it 10? Or is it 1,000? What is enough? So now suppose this is the entire population, the green area. And there was a study, when suppose this green area represents our entire India. Now, the first one, if I say there was a study which was done in, say, state of Delhi, and it has a sample, this big a sample. Now, suppose this big might be 100 or 1,000. Let's take 1,000. Let's say this study 2, was a multicentric study. So we took samples from various parts of India and then we compiled a result. Whereas this study, this was confined to some district in India, one district, and then say Aligarh, and then it was done and results were compiled. So now say this was done in some particular state. So if there are some factors, if there are some ethnic differences for that drug in that state and different states, then this result won't be generalizable because it is representative of that state itself. The second one, when I told that it is a multicentric study, so there what happens is we have different we have representations from different states or different places of India. And there what happens is that is, if we compile the results, then what will happen is we are able to say that this can represent whole of India because we have representations from all over the country. The third one, that is just a district. So now it won't be, say, representative of entire entire thing, because this is just one district and it can't say that if whatever results we are getting, if it would be representative of an Indian population or not, because that is very less amount of, or say very confined population we have taken. Again, I ask you the same question, how much is enough? So what happens if I do my study on a very small number of participants? So even the most rigorously executed study, it might fail to answer its research question just because we have very small number of participants. So we might not be able to capture all the variations, so we might not be able to capture everything. So it might fail. The second thing is it might fail to detect important effects or associations because we have not taken the required number of participants in the study. So again, there might be a failure. And third would be that even if it is capturing that, the measure which is capturing, it is too imprecise. Meaning that it is not capturing what is supposed to measure. Maybe we took some patients which had very severe disease. We could not consider moderate or mild cases, something like that. And then whatever associations we measured were too imprecise. Now what happens? Suppose I talked about if I take very small sample. Now what happens if I take too large a sample? Say 100 were required, I took thousands. What happens? Now my study is becoming difficult. And of course, resources are very limited in our setup. So it is very costly to carry out a clinical trial, and it becomes costlier if you have gone above your required sample size. Second thing would be because you have made affairs costly, it is waste of resources, and it might even lead to loss in accuracy, because what happens is if a sample size is too large, we say that you can prove anything. If it is too small, you cannot prove anything. And if it is too large, maybe you can prove anything you want to prove. So there's a loss in accuracy. So I have been telling sample, sample, sample. So we are not doing with population. Now we are doing with samples. So now what is that sample? That is basically a set of participants or data that is part of the population. It is selected from the population. It is lesser in number than the population, but it would be adequately, like here, if we see there are different subgroups of this population, and we've tried to include something from all the groups, and this participant group represents adequately the population from which it is drawn. And therefore, when we are making some inferences about that population, we are correct in making those inferences. When we say that we are getting some difference or we are not getting some difference, so those things are almost correct. So now, when we are calculating this sample, how much is this sample, we have two approaches. One is precision approach, and the second one is hypothesis approach. So precision approach essentially is where you consider the confidence interval. You must have heard about this term confidence interval, or you must be knowing about this term confidence interval. So when we try to calculate sample size based on confidence intervals, that is a precision approach. Second approach is hypothesis approach, which we follow majority of the times. We follow hypothesis approach only. And when I talk about hypothesis approach, I think I should once revise about what is a hypothesis. So basically, there are two types of hypothesis when we are trying to conduct a clinical trial. We have a null hypothesis, wherein we say that there is no difference. And there is an alternate hypothesis where we say there is some difference between the two groups which we are comparing in that trial. Two or more groups, basically. Now, for this alternate hypothesis, we have one-tailed and two-tailed things. One-tailed is we know which way, or we define which way should the measurement, or which direction should our effect go, so that we say that it is success. So basically, when we say it should be superior or it should be non-inferior, so we are now defining, we are limiting our thing to just one direction. And in two-tailed, it is like we are just saying that it is different from one another. We are not defining which one is better, which one is worse. We are just telling that there lies some difference. What happens is when we do sample size with one-tailed hypothesis, sample sizes, some studies sample size could be 100, in some studies it could be 1,000. Maybe both the studies were comparing those two drugs itself. Suppose a drug X and drug Y were being compared. Still, sample sizes vary in the two studies. Why? So, the first thing is you want to know how precise do you want to be. That means how accurately do you want to measure what you want to measure. It means how much of a factor of chance are you considering. If you want to leave it to chance, like okay, I'll take a 5% chance that my study results will not be just because of the intervention, that might be because of any other thing or by chance or something like that. So if I want to be very precise, no, I don't want to take any chance. I want to be like 1%. I'll give just 1% probability for by chance. Else, I want my study results to be very precise, very accurate. Then sample size will increase. And if we want to be just fairly precise, if we are doing a study, we don't want to be imprecise. So if you want to be fairly precise, the sample size will decrease. This thing represents 0. So here, this side, sample size tends to decrease, and here sample size tends to increase. The second factor is how sure do you want to be of your answer? Meaning, if I have got a result, how sure I want to be that is a true result? If I say that null hypothesis is rejected or alternate is accepted, so how sure I want to be of that answer? If I want to be very sure, then again sample size increases. If I want to be fairly sure, it's fine, I can take like a power of 60-70%, then what happens is I give it to like lesser sample size. The third factor is how much variation is there in the population you want to study. So how much of variation can I afford? If I am taking a spectrum of a disease, say from mild to severe, of course, I am taking a wide variation, and the effect which will appear in those participants would be very widespread. So if I am taking a high variation, if I can afford a high variation, my sample size will increase. If I restrict my population to a very low variation, I take only mild cases, I take only effect size of 1 or 2, something like that, then my sample size decreases. Then the fourth factor is how small of an effect do you want to be able to identify? Meaning, now suppose if we are testing some antihypertensive drug and I want to be I am very cautious and I want to measure even if my drug is better than the other drug in which it decreases the mean systolic blood pressure by 5 millimeter of Hg. And I want to be able to detect that difference. Versus if I tell that, okay, if there's a difference of 10 or 20, then only I want to detect. So if I say that I want to be able to detect a 5 millimeter Hg of difference, my sample size increase. If I say that I am able to detect a moderate difference, fine, 10 to 20 only I want to detect, then sample size will decrease. So summarizing these four factors: effect size, standard deviation, type 1 error with precision. It is by convention is taken sometimes 5%, sometimes 1%, and type 2 error, which will be like, which it will tell about power. So power of 80% or power of 90% is taken conventionally. So these factors basically influence sample size, and we need these factors for calculation of sample size. Now, the consideration in sample size, as you already saw, the first thing would be study design. So this is a holistic thing, like if our study is a cohort study or it is a case control study or it is a clinical trial as we are doing. So we first should know about the study design. Second thing is our hypothesis. If it is a point hypothesis or it is an interval hypothesis, null or alternate as we already discussed. Then what is the objective? Do we want to show equality, superiority, equivalence, or non-inferiority? So we should know this because this will be handy in calculation of sample size. Then we come to our primary endpoint, if it is continuous or categorical, because whatever formula or whatever charts we are using, those will differ depending on our selection of primary endpoint. And the important thing is clinically meaningful difference. As I told you in the last slide, that how much of an effect do I want to measure? Now, I can measure a difference of say 1 millimeter of mercury as well. But then the thing is it clinically meaningful? If it is not, then there's no point in measuring that and telling that my drug is superior or non-inferior or whatever. So that difference which we ought to measure should make some sense, should be clinically meaningful. And last but not the least, if our data which we are trying to collect is normally distributed or not. Most of the times we assume it will be normally distributed, and then only we calculate sample size. So now you might ask, where do you get that effect size? Like, where do you get that clinically meaningful difference and standard deviation from to calculate your sample size? So mostly if you're working in the same area, in the same disease, then you can take it from previous literature, that you can go and check on PubMed or any other such search engine, and you can see if there are some drugs which were acting on that disease, on your outcome measure, that scale or score, and you can check if they have defined anything. So you can take standard deviation and effect size from there if you want to go with that literature. Second thing would be a clinical significance. Basically, if you think that a difference of 10 millimeter of mercury makes a difference clinically, then you can take that as an effect size and you can take standard deviation from previous literatures. The third thing, if the area is entirely new, then you should conduct pilot studies, and then from those pilot studies, which would be done basically based on feasibility. Suppose in 1 month you get 40 patients, you want to do that pilot study for 1 month, and you do that on 40 patients, and then you get some effect size and standard deviation, and then you can just put it in your calculators and you get a sample size. If you have, say, there are two groups and you are getting sample size of two, standard deviation of two groups in from previous literature, you can calculate a pooled standard deviation using this formula of SD square plus SD1 square plus 2 square divided by 2 and everything root over. So you can calculate a pooled standard deviation and you can use it for your calculation. Now, what are the ways to calculate this sample size? So there could be three ways. We have formula, we get that from net, we get that in our textbooks. So we can just put in values in that formula and we can get a sample size. Second approach is, it is not followed now because nowadays we have many software and online tools. So we have some ready-made tables. I'll show you one of those tables. So we have some ready-made tables and wherein you put your effect size and standard deviation and you go to those rows and columns and you get an approximate sample size. The third one, which we follow very widely, is software or online tools. So we have a few softwares and online tools like powerandsamplesize.com. We have Nmaster software. We have IPITSPAS. So we have all these softwares and online tools and we can calculate sample size if we get data regarding the say effect size or mean difference, standard deviation, proportion, whatever we are calculating. If we get those things, we can calculate based on these tools as well. So now straightforward, I'll come to an example. Suppose a clinical trial was planned to study the effect of lowering sodium in diet on systolic blood pressure. So now we have two types of diet here. So during a pilot study, it was observed that standard deviation of SBP in one community, which were taking a low sodium diet, was 11.3 millimeter Hg, while with high sodium diet, it was 13 millimeter of mercury. Sorry, it should be capital. So if alpha is 0.05 and beta, part of the study 0.10. So if beta is 0.10, part of the study becomes 90%. Part is equal to 1 minus beta. Beta is type 2 error. How many persons from each community should be studied if one wants to detect a 3 millimeter mercury difference in systolic blood pressure in the two communities? So how do we approach this? We'll approach this via all the three methods, which I told you. We'll first do it with a formula. Next, we'll see the table. And third, we'll do it with the online tool. Now, for your reference, this is a value of Z alpha by 2 and Z beta. We will be needing it in our further calculations. If you want, you can calculate with me so that basically you get a know-how. So now for a study, for any study, this is the formula for calculation of sample size when you have mean difference in your deposition. So this formula says NC means sample size of, suppose this NC is control group and T is treatment group. This is 1 plus 1 by K, where K is the allocation ratio into sigma square, which is standard deviation square. And these two Z values, 1 minus alpha by 2 and 1 minus beta, alpha by 2 because we are considering a two-sided hypothesis, two-tailed hypothesis here, divided by mean difference, the whole square. So now we did not talk about allocation ratio in that problem. So let me assume that allocation ratio is 1. Other two things which we have with us, low sodium diet standard deviation, that is 11.3, and high sodium diet, that is 13. So we will calculate a pooled standard deviation with 11.3 square plus 13 square divided by 2, and to whole we'll do a root over. That will yield a standard deviation of 12.17. So now we have everything ready as per this formula. Let us put everything in the formula. As I showed in the table in the last slide, Z1 minus alpha by 2 is 1.96. 1 minus beta is 1.282. Mu from the question itself, it is 3. And standard deviation as we calculated, it is 12.17. Allocation ratio was 1. So while I put this into formula, what will happen is it will be 2 into 12.17 square, and then these factors added and divided by 3. So we get a sample size of 345.92 per group. Round it off, we tell 346 per group. So this one is 346. Now in the treatment group, it would be K into NC. K is 1 here. So 346 in second group as well. So it comes to 692 for the entire study. If we solve the same problem via this table approach. So in this table, you see we are taking E by S, that is effect divided by the standard deviation. So what was our effect? It was 3, and standard deviation was, say, 12. It was 12.17. Let me take 12. So 3 by 12, 1 by 4, it will come as 0.25. Now, what did we take? We took a two-tailed alpha, that is 0.05, and our beta was 0.10. So when we come from 0.10 and 0.25, it comes as 336. So it is also approximately similar to that 346 wala sample size which we got from our formula. The third approach could be we go to a calculator. Of course, I'll demonstrate this after I end my slides. So here we put two means which have a difference of 3, and we put our standard deviation 12.17. Our allocation ratio was 1, and when we press calculate with power of 0.9, because our beta was 0.10, so power becomes 1 minus beta. It's 0.9 and alpha is 5%. So it comes as 346 again, which we got from our formula as well. This is also per group. It is NB, if you can see, and here NA by NB is written. So when you calculate NA, it would be K into N of B, and that will again come as 346. Now, we have all these formulas. So what I applied here was a formula for equality. If we have to calculate non-inferiority, we have to add that margin to the denominator. If we have to calculate equivalence, we have to again put this in denominator. If you have to put superiority, this again comes in denominator. So depending on whichever type of hypothesis we have, we can try and we can calculate sample size as well. In our calculators, basically, we'll get all those options. If we have to calculate non-inferiority, equivalence, equality, we get those options and we can go and simply click some buttons and calculate. The second type of thing which we can do in a clinical trial is work with proportions. So there, suppose we take on this problem where I say the response rate for a standard treatment is 60%, and the new treatment would be considered superior if it offers at least 20% more over that 60%. What is the minimum number of patients required in each group to determine whether this is significant at 5% level if we wish to have 80% chance of detecting the difference if it is real? So May I ask, do you get it that this is the power of the study here? Because we wish to have 80% chance of detecting the difference if it is real. So this 80% is the power, and level of significance is 5%. Here, for a control group, suppose the response of treatment is 60%. So for the treatment group, it is 80%, at least 20% more over 60%. So in two groups, we have a response rate of 60% and 80%, which we are considering, and a power of 80% and significance level of 5%. So this is the formula for proportions. And this is from the table 1.96. Now, last time we had this as 1.282 because we had taken power as 90%. Here we are taking power as 80%. Then proportion of the treatment, which we are assuming that it should be 20% more than the control, that is 0.8. And for the control, it is 0.6. This difference wala factor, that is 0.2, 0.8 minus 0.6, that is 0.2. And now allocation again, this is allocation ratio. And as we have not mentioned anything, so it should be 1:1. Now, when we calculate all these things, we come again to a proportion, a sample size of 80 per group. So in control group NC, it is 80. So if we calculate for treatment group, it would again be 80. So in this study, we will take a sample size of 160. Now, again, going by this table approach, it says here the smallest of P1, 1 minus P1, P2, and 1 minus P2. So I think amongst all these things, smallest is 1 minus 0.8, that is 0.2. So let us stick to this 0.2. Again, P2 minus P1, that is also 0.2. So this one 0.2. And when we match this abscissa and ordinate, we get this as 82. We got 80 from formula, we are getting 82 here. Next would be our approach of calculation using a calculator. And here, group A proportion was 0.6, group B was 0.8, sampling ratio is 1. When we calculate, we get a sample size of 79 if we assume a power of 0.8 and a type 1 error rate of 5%. The other thing which we should take into account is a dropout ratio or dropout rate, what we apprehend. So if we are apprehending a dropout rate, then what we should do is we should also include that factor in our sample size calculation. And suppose here we got a sample size of 79 or 80, and I assume a dropout rate of say 20%, then what I should do is I should do 80 divided by 1 minus 0.2. That would be 80 divided by 0.8. And then I should comment on my sample size, which will come as 100 now for one group. So for two groups, it would be 200 then instead of 160, which we just calculate using the formula. So basically, for any study, a priori sample size determination is to be done. And level of significance, power, and effect size has to be considered if we are calculating any such say sample size. Now I would like to show you this calculator. So here, if you can see on my screen, basically you can see that it is powerandsamplesize.com. This is the say site where I generally use for calculation of sample size. There are other sites, there is EpiTool, there are PAS and all those things. You can use any whichever you are comfortable with. So as we open this window, we generally come to this page. We can say here, go straight to calculator. We can click this. Go straight to calculators and it does come. Then you see it is test 1 mean. Here, not in a clinical trial, but we can do it in cohort study or a cohort study basically. This one says compare two means. This we can do for a clinical trial. Now here you see two sample, two-sided equality. Two sample, one-sided. Here, 2 sample non-inferiority or superiority and 2 sample equivalence. So as I calculated using equality, I'll show you this. Now, it asks for certain inputs. You can adjust power of the study. Here you can change it accordingly if you want 80, if you want 90, or if you want lesser. You can adjust type 1 rate, 5%, 1%, whatever you want. Then what you have to enter is the mean of 2 groups. Now, what we saw in that problem, I had already given that there is a mean difference of 3. So I'll show you one thing. See, if this is 5, to get a mean difference of 3, what we can do is we can just add 3 and put it here. The mean difference becomes 3. We had a standard deviation of 12.17. And we had an allocation ratio of 1 only, and we calculate sample size. It comes as 346 as we did earlier. Now you might want to ask, why did I take 5 and 8 here? The thing is, for this mean difference calculation, you can take any number of things. Like it is 8. Now suppose if I take 11, mean difference is again 3 only. And if I calculate, sample size does not change. It is 346. So if you have got a mean difference, you can put those values which might give you a mean difference of that number, and you can go ahead and calculate. What happens, as I told, we were talking about factors affecting sample size. So let us play a bit and see if I do this a type 1 error rate as 1% and then calculate. What happens? Sample size increases. If I decrease power to say 80% instead of 90 and then calculate, sample size decreases. Now, if I increase the standard deviation to 15.17, what happens? Sample size again increases. If I reduce the mean difference, it was 3 earlier, now it is 2, 10 minus 8. What happens? Sample size again increases. So what happens if we increase power? Sample size increases. If we decrease this type 1 error rate, sample size increases. If we decrease the mean difference, sample size increases. If we increase standard deviation, sample size increases. So on increasing power and standard deviation, sample size increases. On decreasing type 1 error rate and mean difference, sample size increases. So if we want to get a lesser sample size, or if we want to adjust for one of the things, we can play with these things, given that doing those things are ethical at that point of time. We are not just doing to decrease sample size or something else, right? We'll again see those calculators. And here you can see this is a 2 sample non-inferiority or superiority. So let us see that once. So it is similar to whatever we saw earlier. Here, thus, there is just a factor of non-inferiority or superiority margin, which is added to the whole thing. So we'll go with our earlier example and let non-inferiority margin be 2 and then say what is the sample size. Say it is 74. So now with the same thing, we are getting a lesser sample size here. Then if we go to say proportion. So test 1 proportion. No, we don't do this in clinical trials. We can compare 2 proportion with 2-sided equality as I did in other example. Now we had 0.60 and we had 0.80 and we had to calculate sample size. It is coming again as 79. In our example earlier when we did with formula, we got 80, so it is similar. Now, as I told you in mean difference, that you just have to maintain the difference and see. That is not feasible with proportions. Now the difference is 0.2. And suppose to maintain this difference, I do this as 0.6 and 0.4, the sample changes. So here, when we are specifying proportions, we need to put the exact values what we had. Like if we have a baseline rate of say 0.6 and we're talking about a 0.2 over and above that rate, then it means it has to be put as 0.8. If we are talking about below that rate, then only we can put it as 0.4. So this is the major difference. While you are calculating with mean difference, you can put any two values which maintain a mean difference as specified. And in proportions, you have to put the exact proportions, because if you just change these values, your result will change. So you should put the exact proportions. The other things would be like if you want to compare many groups, if you want to compare paired proportion, if you have some time to event data. So if you see the time to event data, now here also it asks for a hazard ratio and overall probability of an event. So now suppose if I consider hazard ratio of 0.65 and overall probability of my event is 0.8, it asks for proportion of sample in group A. So 0.5 means that our allocation ratio is same. In group A, it is 0.5. In group B also, it would be 0.5. So we get a sample size of 212 here. So these could be the most used approaches while we are talking about calculation of sample size. Or you can, you're most welcome. This is a free website. You can go and you can basically play with all these things, all these numbers, and you can calculate sample size for your study and for other studies as well. So with this, I think I finish my slides. And thank you so much for your patient listening.
Thank you so much, ma'am, for your engaging presentation. And now I will ask the questions from the delegates now.
Yeah, sure.
Ma'am, the first question is, what is the role of ethics committee in determining the appropriateness of the sample size with regards to the protocols they check? Over to you, ma'am.
Yeah, actually, ethics committee is not so much concerned about your calculation of sample size. They would just be checking if you have taken too much of a sample, or the method you have used, does it apply to your, like if I am doing a sample size calculation for say mean difference and I take a formula for proportions, so those things would be evident. Else, if it is within bounds of that, then ethics committee is not concerned about your sample size thing if you have used the correct formula. And if you're not talking randomly that I'll use a sample size of 100 for this study, so then they'll ask you to calculate and show the calculation.
Thank you, ma'am. And the next question was sample size for pilot studies. Any minimum number is there, ma'am?
Actually, for pilot studies, basically they take 30 to 40, but it then again depends on feasibility of the studies as well. Because like recently, we have all faced a pandemic of COVID, and many people who started on feasibility, they got a very less sample size than they assumed. So pilot studies mostly work on feasibility. Now, see, if you're doing a pilot study on diabetes, you can easily get 40 to 100 participants, right? But if you are doing a pilot study on some oncology drugs, then what will happen is you won't get those many patients. So it will mostly depend on feasibility.
Thank you, ma'am. And one final question, ma'am. Can you explain an example with how to, based on type 1, type 2 error and SD or determinants for sample size calculation? And if there is any statistical tools to calculate sample size for animal study in my thesis?
Actually, I'll take the last question first, that there are no statistical tools for calculation of sample size. However, there is a sample size thing, like if you should have your number of animals minus number of groups between 10 to 20. That is the best thing to do. So you have to do it like that only, because no tool, I don't think so there's any tool. If there is any, it is, anyone else can answer this. But in my knowledge, there is no tool. Other thing which you asked, that with an example, so I think I explained 2 examples. Those were based on type 1, type 2 errors. Type 1 error was 5% there, and type 2 error was 0.10 for first and 0.20 for the second one. And standard deviation we already took as 12.17. Mean difference we are taken as 3, and then we calculate the sample sizes. So I think I have taken this, maybe this question was asked before I went to the example, maybe.
Thank you so much, ma'am. Thank you, ma'am. Thank you for your time and your valuable presentation, ma'am. And if there are any questions, they will be posting on the chat box and we will post the answers in the group.
Yeah, thank you so much.